All right, so we're going to be talking about muscles, but more from the microscopic level. So talking about the proteins and all those small structures on the inside, which can be kind of hard or complicated to picture because, well, they're microscopic. So I want to kind of start zooming us in. We're going to start with a whole muscle and talk about how it's organized and work our way down to those small little proteins. So looking at the whole muscle, it's giant. These cells have actually merged from multiple cells, so they're actually not even called cells anymore by the time they get this giant. We call them fibers. And so we have these giant cells grouped together in bundles, and then we're going to take those bundles and then group those into bundles, and that's what makes a whole muscle. So we'll take a whole muscle and we're going to group them up into fascicles. Fascicles are then just the groups of the cells themselves, which remember we don't call cells, we call fibers. So we have a muscle fiber. So you're looking at a muscle fiber, so if we were to try to draw this out, it would be like one cell. And a fascicle is going to be a group of those cells. And these are all going to get wrapped around in coatings. So around the muscle fiber, there's going to be one little coat. And around those fascicles, those each get a coat. So here's our fiber with the coats around them. And then we'll have the fascicle that'll have a coat around that. And then the whole muscle is a whole bunch of those. So now we're going to have a bunch of the fibers on the inside of those for the groupings. And those have their coats around them. So kind of like that. So the analogy I use is you think about okay, you're holding a pencil. That's like your fiber. And around that, outside of that pencil, is a little plastic coat. That little coat right here that I'm drawing in our orange color, that's the endomysium. So the endomysium around each one of those fibers. Now we're going to take those pencils and we're going to wrap up a group of those pencils with a rubber band. That would be what I'm drawing up here in pink. So this is going to be the perimysium. So that's what's surrounding a fascicle. I guess I'll do it like a bracket so you can see it's surrounding it, it's wrapping around it. So we have a muscle fiber that's wrapped in endomysium, and around the whole muscle, of course, we're going to put a coat around it because we cover everything here. We're bundling with bundles. I have bundles. And so I'm going to do an epimysium around the whole thing. So that's kind of how we're grouping them. We have bundles inside of bundles inside of bundles. And so again, going back to the analogy, the perimysium is a group of pencils with a rubber band around it. We're going to take a couple of groups of pencils and we're going to put them into a pencil box, and that's our whole muscle. So the plastic coat around that pencil box, that would be the epimysium. Hope that makes sense. We even keep that same plan once we get inside of the cell itself. So inside of the fiber, we have, you guessed it, bundles. So those muscle fibers are going to have bundles called myofibrils. Myofibrils are also surrounded with things, but not connective tissue coats like these. These three are all connective tissue coats. On the inside, how the myofibrils work is you're going to wrap them around with extensions of the cell membrane that allow the electrical signal to get deep into the fiber. That's a transverse tubule. So we have a tube that's coming in, and it's going to wrap around these bundles. So we'll have transverse tubules. And then between those transverse tubules, remember this is about getting the electrical signal deep into the muscle fiber, we're also going to have something called a sarcoplasmic reticulum. And that's going to allow us to take that electrical signal and turn it into a chemical signal that actually can talk to by binding the proteins that are actually going to do the job of the muscle contraction. So speaking of those proteins, let's talk about them. So myofibrils are bundles of these, what we call myofilaments. Myofilaments are going to go into two main categories. You're going to have your thick filament, and you'll have your thin filament. With the thick filament, these are the ones that actually do the pulling in a muscle. These are going to be made out of this protein called myosin. They look like golf clubs. So we have this long part and a little head that sticks off the side. And so if you're thinking about, like, here's my thin filament, here's my thick filament, the thick filament reaches up with a little glove-like head, pivots towards the midline, and it'll keep doing this, and it will shorten by pulling parts of the muscle towards each other. So we'll do the same thing on this side. The head will reach up and it'll pull it in. So we're going to pull these two parts towards each other, and we're actually going to cause some overlap that allows it to shorten. So what we're going to have with the myosin are the heads that reach up and pull. So it means the thin filament must have the thing that it can grab a hold of and pull on. And so the primary protein for this is actin. Actin will look like a little ball. And there's actually a whole bunch of them. We're going to put them in rows. And we're even going to have 2 rows of them right next to each other. And to help hold them in position, there's actually a rope that goes underneath. And that's called nebulin. Now, we don't want the heads of the myosin to be able to grab a hold and pull at any time. We only want them to be able to do that when it's time for a muscle contraction. We know that because the signal has been released from a neuron and travels down that transverse tubule to get the sarcoplasmic reticulum to release a chemical message, which, by the way, we haven't even talked about yet, is calcium. So we have to have something on here to respond to that calcium and something on there to hide where the myosin would attach. So let's go through those two. So the first thing that we'll have is going to be the rope that covers the myosin binding sites, and that's tropomyosin. So it's a rope that sits over top of the myosin binding site. So myosin binding sites is where the head would attach to be able to pull on it. So then you're going to cover it with this rope. So the way I remember it is I think tropomyosin is trouble for myosin. It's in myosin's way. It's blocking the binding sites when the muscle's at rest. It does have little handles built onto it. And so we have these little handles that will pull on tropomyosin. So it's our troponin. So troponin are the little handles that will move it when calcium binds to it. So remember that calcium we were talking about a minute ago? That's what's in the sarcoplasmic reticulum. So sarcoplasmic reticulum releases calcium, and calcium can bind to that little handle. When it binds to that handle, it's going to pull. And when it pulls, it's going to move the tropomyosin out of the way, and now the myosin heads can grab the thin filament and pull on it. So that's why understanding the organization of all this really is important, because it really helps you tell the story of how a muscle does a contraction.